name is Jonathan Bibliowicz. I'm Associate Director of Cancer Research and Oncology for PacBio. And some of you might know us as, in the world of sequencing as long-read sequencing. There's short-read sequencing, which is the traditional sequencing. I'll explain in a second the benefits of it and show you a couple of examples here we go, of how we utilize long-read sequencing and why it's important. But before that, this year has been really a seminal, a really important year in genomics, in sequencing especially. Two things happened at PacBio. One, we finally launched a sequencer that can really handle the genomes and transcriptomes and the depth of sequencing that is needed in cancer genomics. So really excited for that, and I'll show you some kind of ideas, preliminary ideas of what we can do with this technology. And at the end, I'll show you our new sequencer we just launched and it started shipping, which is the world's most accurate short-read sequencer. So we have the most accurate long-read technology and the most accurate short-read technology. In the world of cancer genomics, you can imagine that is incredibly important. The accuracy is really important. So on the right is the Revio system. That is a very high throughput system we just launched this year. And basically it's able to handle somatic genomes, transcriptomes, and epigenomes. So we're really looking at the full multi-omic landscape of tumors. And I'll show just a couple of slides. I have just a few minutes, unfortunately, but you can always stop by our table and get more information. To back up, this is a publication for Mike Schatz from at the time at Johns Hopkins that really showed us as an example why long-read sequencing is so important in cancer. As we know, cancer genomes are highly rearranged. And when we use short-read sequencing, because these structural variations are so large, we use short reads, we aren't able to really kind of figure out what's going on with this major large changes within the genome. So we use these very long reads and taking, for example, here, sequencing a well-characterized, quote-unquote, cancer cell line that was thought to be well-characterized with short-read sequencing, found 20,000 new structural variants with our technology. This is about 5 years old, but one of the first examples of the power of really sequencing very long fragments to understand cancer genomics. So with this Revio system, we can now do very high throughput genomes and at very high depth and very cost-effective. This is just a fairly new data of a cancer cell line, for example. So you see the reads of the tumor on the top and the normal. In the tumor, you see this deletion, this structural variation. And on top of that, you see the blue and the red, and that's methylation, right? So we're able to, within the same read, get methylation for free, which allows us to do what we call 5-base sequencing. I know it's a lot of information, but the 4 bases plus methylation for free. So we're able to start to understand the interplay between the genomics that drives cancer, but also then the epigenetics that result from these changes. So on the right, you have this deletion. On the left, you have hypomethylation in the tumor, which you don't see in the normal, in the p53 pathway. So again, starting to leverage this technology to really understand the complex sort of multi-omic dynamics of cancers. But the majority of our focus, at least this year and last, has been in the world of RNA. So we know that cancer is a disease of the genome, it's driven by mutations, but a lot of the dysregulation happens at the level of RNA. So things like RNA isoforms, fusions, which have been talked about widely, gene expression, obviously, with RNA, but also express mutations to look at tumor evolution, things like neoantigens. So what we're doing, and I'll show this example here, using long-read sequencing, we're able to capture all of these variants without missing any information. So again, I'm running out of time quickly, but on the left, you have an example of what happens when you do RNA sequencing using short reads, just traditional sequencing. So you get these short fragments. A gene or a transcript is about 2 to 2.5 kb. These fragments are about 200 base pairs at most. So you're able, with these short fragments, sort of like a puzzle, assemble the transcriptome, about 20 to 40% of it. So you're missing a lot of information of the large stuff, the isoforms, the fusions, the structural variation on the genome. So you're getting really a partial view of the transcriptome. We're able to sequence RNAs from 5' to 3', right? So we're sequencing the whole molecule as is. We don't have to reconstruct it. So we're really getting a complete view of the transcriptome, getting things like RNA isoforms, fusions, and because of the accuracy, we can really detect point mutations. So really getting a view of what's really going on, which I think with our traditional tools, we've been missing a lot of. Really quickly, one great example from Science Advances from last year. This was done at Jackson Labs in Connecticut. Did this comparison for the first time in different types of tumor samples, short reads versus long reads, and identified, in this case, tens of thousands of new isoforms, RNA isoforms that were not known before. So one of the first sort of big atlasing projects that showed us that we're missing, in this case, about 2/3 of the information when we use the wrong technologies. And we've taken this, we partnered with 10x Genomics, which many of you know. We can now do this at the single cell level. So we can use a 10x Genomics platform, isolate single cells from a tumor or any tissue, and perform this long read sequencing on single cells. So this is fully supported, and you can actually buy a kit. And this is just from the original publication, looking at this heterogeneity of cell types within a tissue, and really trying to understand not just the gene expression. For those of you that have done these studies, you're really looking at gene expression, but then on the right, really start to superimpose the isoforms, the fusions, the mutations onto this data, and understanding more of the underlying biology at the level of RNA. So if you have any questions on this, please stop by and you can talk to my colleague and myself. So I'll skip a couple of things because I just simply ran out of time. But what I want to really focus on, if you look on social media and you follow us, we just announced the first shipment of a new platform, a short read sequencing platform. We now work in both worlds. This is a benchtop, sort of mid-throughput. If you know of an NextSeq, this is sort of equivalent, very equivalent, that does all the things that an Illumina sequencer would do. But on the right, you see that our accuracy is 90% over Q40. So for those of you working in the world of sequencing, that is very impressive. And what this means is that when you look at our Q-scores, or really accuracy scores, that's in magenta, and then SBS, which is the predominant sequencing technology on the market, in orange, we're 15 times more accurate. So when you start looking at the data appears to be almost error-free. We're not error-free, but we're close to being error-free. And again, in the context of mutation detection, it's a very similar technology, just more robust and more accurate. So I want to end with two figures that impressed me when I joined PacBio about 1.5 years ago, and hopefully will impress you. And this, again, this platform is now out and being used. The first one is, this is a comparison from the world of liquid biopsy. In liquid biopsy, as you know, the fragments that are floating in the blood are very short. So you don't really need long read sequencing. There are at the most 200 base pair on average, so you can get by with a short read sequencer. And we did a comparison with the SBS, the current leading technology of sequencing, and our technology to see what do we get when we do a liquid biopsy test, and what does the accuracy mean. Here you see an IGV plot, so you see all these reads. On the right, this is the SBS, the predominant sequencing technology on the market, and you can see the true variant on the right that's marked, and all of these colored bases around. Those are errors, right? So what happens is when you have on the bottom right, the true variant, which in this case is a C, which is what we call the signal. And then you have all these errors around the signal, that is noise. So the noise is overcoming the signal, and you can't really tell what the true variant is. You have a really hard time detecting what is the true variant, in this case in the liquid biopsy test. When we use close to error-free sequencing on the right, you see the errors are gone. So the reads are clean. And the signal, which is the C base, we know that is the true variant, rises above the noise. You see A's and G's are gone, they're zero, the errors are gone. So the way we envision this technology being used is in the case of rare variant detection, in things like liquid biopsy, but also any scenario of rare variant detection, where you're really looking at really low variant frequencies, and you need very clean, very robust, very accurate technology to detect that variant consistently. We've done more and more studies on this, and on the average, the sensitivity of these assays is about twofold what we get with just the same library, same sample, using current short read sequencing. So again, I think this is really exciting and really the future of short read sequencing, and we really hope that some of you will stop by and maybe have some project ideas or any questions around this technology. But again, this technology is now available on the market. So with that, I'm out of time, but thank you very much, and please stop by and we can have some more conversations.